good afternoon, and good evening, everyone. Welcome to today's lesson on RNA structure. I am Christine Faith Blando, and I would be your teacher for today's lesson. But now, let's start with a little warm-up. And I'm going to show you 4 pictures. Yes, you heard me right, 4 pictures. Here we go. Now, we have a ribbon, a sugar, the letter U, and a virus. Now, think for a second. What do you think these pictures have in common? Well, if you guessed RNA, you're absolutely right. Each of these represents something important about RNA structure or functions. That's exactly what we're going to talk about today. Let's take a look about our learning goals for this short session. By the end of this lesson, you should be able to identify the key structural features on RNA, meaning its single strand, ribose sugar, and uracil base. Next is to compare RNA with DNA, especially focusing on ribose versus deoxyribose and uracil versus thymine. Thirdly is to understand how RNA folds and what makes it flexible. Fourth is explain RNA's role in transcription and translation. Fifth is to evaluate how mutations in RNA can affect cell function and health. Sounds like a lot, right? But don't worry, we'll take it step by step. Alright, let's begin with what exactly does RNA do in the cell? RNA actually plays many roles. It doesn't just sit around, it acts. It's the messenger, the builder, and the translator. There are 3 main types of RNA that you'll encounter. First is the mRNA, or the messenger RNA. It carries the genetic code from DNA out of the nucleus to the ribosome. Next is the tRNA, or the transfer RNA. It brings amino acid to the ribosome, while rRNA, or the last one, which is ribosomal RNA, it helps form the ribosome and is essential in assembling proteins. Well, these types of RNA work together during protein synthesis, which we'll also explore a bit later. Now let's move on. How is RNA different from DNA? Here's a simple way to remember it. DNA is the blueprint, and RNA is the worker. The DNA stores the information while the RNA carries out the instruction to build proteins. Let's compare them side by side. DNA is double-stranded, while RNA is single-stranded. DNA has deoxyribose, while RNA has ribose. DNA uses thymine, and RNA uses uracil. DNA stays in the nucleus, while RNA moves in and out. Understanding these differences helps us appreciate what makes RNA uniquely suited for fast, temporary jobs in the cell. Now let's zoom in on one of RNA's key structural features. RNA is single-stranded, and this is a big deal. Think of DNA as a twisted rope. It's strong, stable, and structured. Now, think of RNA as a ribbon. It's light, flexible, and bendy because RNA is single-stranded and it can fold into all sorts of shapes. And this flexibility allows it to perform many functions, including acting like an enzyme in some cases. Now, let me ask you, why do you think RNA needs to be flexible? Great! The flexibility helps RNA fold and interact with other molecules during transcription and translation. Now let's talk about sugar, literally sugar. DNA contains deoxyribose, but again, RNA contains Ribose. Here's the difference. Ribose has actually one more oxygen atom than the deoxyribose. And this small change makes RNA more chemically reactive. And that is why RNA is usually short-lived and perfect for temporary instruction. You can think of it like this. Ribose makes RNA like a fast, lightweight delivery bike. It gets the job done quickly and moves on. Now, let's take a look at the bases, specifically uracil versus thymine. Well, DNA uses adenine to thymine, A to T base pairing. But have you wondered RNA? It actually replaces thymine with uracil. So now we have adenine to uracil, AU pairing. But why the change, miss? Uracil is actually smaller and more suited for quick pairing and breakdown. Also, enzymes can recognize it as a sign that the molecule is RNA and not DNA. Now, here's a quick quiz for you. What base pairs with adenine in RNA? That's right, it's uracil. Because RNA is single-stranded, it can fold into complex shape like loops, hairpins, and bulges. Let's use an analogy. Imagine a flat piece of origami paper. If you fold it the right way and you can make a crane, a boat, or a heart, that's what RNA does. It folds itself into shapes that help regulate gene activity or even catalyze chemical reactions like enzymes. But have you wondered, can DNA fold like this? Not really. RNA in transcription and translation. Let's move forward. Let's now put everything together. So how does RNA help make proteins? Well, in transcription, the DNA copied is into mRNA. Then in translation, the mRNA is read by the ribosome to build a protein. But everyone, here's actually what happens. mRNA carries the genetic instruction, tRNA brings the right amino acid, and RNA helps build the actual protein. Let me show you something. This is how life builds itself, from code to action, well, all thanks to RNA. But everyone, let's imagine a real-life situation. You're in a laboratory preparing an RNA samples for a vaccine. Then someone accidentally uses DNA instead. What's the issue? Well, DNA has thymine and not uracil. It's almost more stable and not designed for quick expression. RNA structure, its ribose sugar and uracil base, makes it perfect for temporary expression, which is crucial in fighting viruses like COVID-19. So using DNA instead of RNA, it wouldn't just work. Finally, let's recap what we've learned. RNA, is it single-stranded or double-stranded? Single-stranded, correct. It contains ribose sugar and it uses uracil instead of thymine. RNA is also flexible and folds into shapes. It plays a vital role in transcription and translation. Now, I'd like you now to complete the worksheet provided in the MS Teams, and you'll draw and label an RNA nucleotide and compare DNA and RNA using a Venn diagram. Take your time, everyone, and of course, we almost not forget to finish off this lesson. I'm asking everyone to complete a short 5-item quiz via Google Forms. Only 5 items, and the link is in the description. You have 24 hours to submit your worksheet and quiz. If you have any questions or thoughts, feel free to post them in the comments or chat. Thank you everyone for learning with us today. And as always, keep anti-buddying.